Now that we've talked about DNA's makeup and the different components of DNA, now we have to talk about how DNA is protected. So if you can imagine that DNA is what we said makes you, some of the different components of it make you look like you look, and different components make you act like you act, make you think like you think. So it's important to protect that DNA so it's not altered in any way. And so because that DNA has that special coding that you inherit those traits, it must be accurately transferred. So that means that you have to get the makeup just as it is made inside your body at the molecular level, right? So cells are constantly growing and dividing. And remember when we were talking about cell structure and function, we were talking about how if your cells just built and built and built and built in your body, how would you have all the room for these cells? Just like your cells can't completely multiply over and over and over and over without having room, they also can't break down and die, break down and die, break down and die without being replaced. So they're constantly growing and dividing, and they are growing and dividing without the DNA being damaged. So you can't break the DNA strand as your cells are being replaced. In order to protect the DNA, it's tightly coiled around proteins. Remember, proteins help with structure and building and transport. And the special proteins that help the DNA be protected are called histones. So they are called histones, and then they're condensed into chromosomes. So when you have a bunch of DNA protected with the proteins called histones, they end up being called a chromosome. A nucleosome is a histone plus the DNA wrapped around it. So the histone is the protein, the DNA is all wrapped around it. That makes the nucleosome, and then with the histone, the DNA, the nucleosome all wrapped together, it is what we know as a chromosome. So now we're talking about how is DNA copied. So think about just like with your cell phone, with computer, with your iPad, with whatever, how you are copying and pasting. So when a cell divides, the DNA has to be copied. So it's a copying and pasting process per se. The process of copying DNA is called DNA replication. Before you can replicate, so before you can copy, which is replicate, before you can replicate the DNA, it all has to be, remember how we talked about the double helix, and it's the ladder that's all wrapped together. So it all has to be unwrapped before you can go through the process of replication. So after the DNA is unwound, so it's unwrapped from the histones, which is the protein, an enzyme called helicase. So helicase is an enzyme. Enzymes break down. And so the helicase breaks the two strands down of the DNA. So that's after it has been unwound. So that process is known as the replication fork. So where the helicase unzips the rungs of the ladder, right, is known as replication fork. So these are all words that are important to know, different terms that may be new to you. Another enzyme, so the first one was the histone. Another enzyme is your DNA polymerase. DNA polymerase brings nucleotides to match one of the strands, known as the parent strand. So the parent strand is one of the strands. After you unwrap it, you unzip it at the fork, and the parent strand is your original strand. The new sequence will exactly match the old sequence because of the strict base pairing rule. So we talked about the pairing rules of the nucleotides and how the different components must be matched together, right? So now we're getting down to where we're identifying our parent strand. Your new strand is your daughter strand. So depending which portion of your chromosomes you are pulling together to form your DNA strand is dependent on what your parent strand is to what your daughter strand will be. So we will talk more about that, and you will understand that a little bit more. We'll work with it more. But because each strand has its own identity, one's the parent and one is the daughter, because only half of it is the parent in the new molecule, that process is called semi-conservative. So semi is half, And then conservative means, it's what is existing, it's the traditional. So semi-conservative is that part of your DNA. So to make it easy to move DNA without damage, it is wrapped around histones, that's your protein, and condensed into chromosomes. To copy the DNA is called DNA replication. Enzymes separate the two strands and use one side as your blueprint to make a complementary strand. So your parent strand is the one that is existing, and then the daughter strand comes in to pair up, right? Okay, that's all of this section, and then we will keep getting a little bit more and a little bit more as we move into our discussion about DNA, RNA, transcription, and translation. Make sure you're learning all of these terms and understanding each part of this before you move to the next topic.